Thank you very much for the ASA Biopharmaceutical section, Distant Learning Committee, for the invitation for us to present our research at this forum. My name is Weili. I'm from Ivy. And also I have my co-presenter, Mark Levenson, from FDA CEDAR. Today we'll talk about the statistical considerations for the evaluation of fit-for-purpose real-world data. Next slide. So this is the usual disclaimer. Next slide. So let me just in the first few slides to introduce our scientific working group. So what we are presenting is the output from this ASA Biopharm section sponsored real-world evidence scientific working group. So we set up our goals when we set up this working group in April 2018. We want to advance the understanding of the real-world evidence research in a pre-competitive space, engage regulators on providing guidance and/or guiding principles on the real-world evidence research, also facilitate the utilization and implementation of real-world evidence, real-world data in clinical research and the medical product life cycle. Where appropriate, we want to develop statistical methodologies for evidence synthesis and to advance analysis approaches. Because this is a field very much evolving, we would like to educate the broader statistical community to understand and contribute to this important area. We also want to increase our statisticians' leadership role in the cross-functional collaborations in the uptake of real-world evidence and real-world data applications, and communicate a statistical perspective to a larger clinical trial community. Next slide. So our working group, as I mentioned, started in April 2018, so it's about 4 years now. The work presented here is based on our phase 2 effort. From early 2018 to summer of 2020, we had a phase 1 output. We actually had 3 papers, all published in Statistical in Biopharmaceutical Research in early 2021. As you can see on the slides, we have 3 papers on current landscape in biostatistics of using real-world data and evidence for medical product development, which is a general consideration. The second paper is on clinical study design and analysis. And the third paper is on causal inference framework for study design and analysis and analytics. Phase 2 started in summer of 2020. So it's about almost 2 years, and a year and a half actually. So as you can see also, we divided the team by 3 sub-teams. Team 1 looked into estimate from a concept to application in real-world studies. Team 2, which is our team, with Mark and I are the co-leads. We research into the statistical considerations for the evaluation of fit-for-purpose real-world data. And Team 3 showcasing examples of applying causal inference roadmap to real-world evidence clinical studies. So we have submitted Team 2 paper already to SBR, which is the Statistical in Biopharmaceutical Research. And the Team 1 and Team 3 are almost ready to submit in the next few weeks. Next slide, please. So this is a slide on the team, our Team 2 membership. So again, Mark and I are the co-leads. We have the industry representations on the left, and also academics and FDA representations from right, as you can see. So this is outline of the presentation today. So Mark will touch on start by introducing the guidance and the study types. He will also cover the data source examples up to the validating and assessing biases. And then I will take over at that point. We'll discuss our semi-quantitative approaches in how to assess fit for purpose real-world data sources. And then I will wrap up with concluding remarks. Mark?
Okay, thank you, Weili, and thank the ASA Biopharm session for hosting this webinar. So I'm going to start off with some kind of basic definitions and some basic framework on real-world evidence and review some of the FDA guidances. And then I'll basically go through the sections of the paper that on fit-for-purpose data that Weili mentioned a moment ago. So just to kind of get us all on the same page, we're working with FDA definitions of real-world data and real-world evidence. And the distinction is that the data from the evidence derived from the data. So there are various sources of real-world data. Just a few of them are listed on the left. I'll just talk about maybe the first two or three. Electronic health records. So that's basically when you go see your physician and they're entering information into some laptop or something. They enter, their observations. They may order tests and get test results back. They'll note that they gave you a prescription or something. So it's basically where your electronic health data is stored for your physician. Another big source of real-world data are claims and billing data. Probably, if you're not working in this area, you're probably less familiar with this sort of data. These data are generally used to administer a healthcare system, whether it's like U.S. Medicare or some HMO or some insurance. They contain information on billing, but along with the billing are information on diagnostic codes, procedure codes. Included in the billing are prescriptions filled. So there's a lot of information in those data, and although they weren't designed to study like the safety and efficacy of drugs, they've been used for some time to study certain drug questions. And the first two each have their strengths and weaknesses. And our first landscape paper that's already published that Weili mentioned, go through the strengths and weaknesses of each of these data sources. Another one worth mentioning are product and disease registries. Most of you are probably familiar with these, and these could be very high quality data. And again, there's pluses and minuses to these. So finally, there's also things like maybe data that's collected in your home. And this could be something like a smartwatch or other devices that collect data that might inform health status. So with real world data, you use it in some study, and the results of that study are real world evidence. Now, the important thing to note here is the real world evidence is not synonymous with observational studies. That's FDA's position, and that's also been the position in our ASA Biopharm working group, that it includes randomized studies as well. Whenever you use real world data, that will give you real world evidence. So that may be in a randomized trial or such as large simple trials, pragmatic trials, or observational trials. And now I'll go on to go through more details of how you might use real world data in a number of different studies that you're probably all familiar with. So this is supposed to represent a spectrum from sort of a minimal use of real world data to like a real heavy use of real world data. So on the left, we start with the traditional clinical trial. The trial itself may not use any real world data, but before you conduct a trial, real world data may actually, or often is used to maybe design the trial, to understand the disease population, the comorbidities, the incidence of the disease, the incidence of outcomes. So there's a lot of things you might use real world data and then design a traditional clinical trial that may use only like specially collected data, like in a traditional trial. Moving up this ramp, RWE may be used for site selection. And this there's many other uses. This is just examples here, but you might use the RWD to figure out what physicians are seeing patients that you're interested in for your clinical study. Where there might be a lot of patients. But once the sites are selected, again, it might look very much like a traditional clinical trial. Moving up further, maybe you use the RWE to actually select individual patients for your study. So, based on some inclusion-exclusion criteria, you might select some patients, and then they may go through informed consent and screening and additional inclusion-exclusion criteria. But you might use this to actually identify the actual patients that may enter the study. Another step forward is using RWD for outcome data. In my opinion, this is actually a rather large step because it's important to get the outcome correct. You can have a randomized study and good compliance and everything, but it's important to make sure the outcome is a valid outcome, an unbiased outcome. But you may be able to use from medical records or other sources outcome data. And we'll talk further about validating data as well, and that's relevant here. Further up the ramp is perhaps your whole study is embedded in some healthcare system, like the US Veterans Administration healthcare system. So this, maybe you get your medical history from the medical data, maybe all the follow-up, all the adverse event follow-up, all the outcomes are already recorded in the health system. And it's just maybe some randomization at some point of care. Or perhaps most of the data is available in the healthcare system, but there's just some additional data that's needed for your study. Moving on to the non-randomized studies, the external control studies. So you have a single arm intervention, and you might use random real-world data for the external control. And finally, two examples of completely non-interventional studies, a prospective cohort study and a retrospective cohort study where there's no intervention, it's purely observational. And so there are this is not an exhaustive list or sequence of the use of real-world data, but this is just some examples of increasing uses of real-world data. So any study, whether for regulatory purposes or otherwise, should be of high quality or a quality sufficient to address, the research needs. But of course, regulatory studies have to be of a very high quality. And a lot of that's actually described in federal laws, federal regulations, and of course, guidances. And focusing on efficacy for a moment, because I think there's a lot more written on efficacy. In order to get a drug approved in the US, you have to prove it's efficacious. And that's done by providing substantial evidence through adequate and well-controlled trials. So I'm basically, recalling US statute, US law now. But what is an adequate and well-controlled study is described in US regulations. And I'm paraphrasing that here. There are sort of 7 elements to what constitutes an adequate and well-controlled study. And the first one is clear objectives and summary of methods and results. I think that's pretty self-explanatory, but it gets at transparency and clarity. The next one is the design permits a valid comparison with a control. So, people get better on their own, people get worse on their own. And without a control, you don't know if it's a natural history or the effect of the drug you're trying to evaluate. So you need a design that's actually makes some comparison with a control. What's interesting here is the the regulations talk about concurrent controls, so that might be in a randomized study, but it also mentions historical controls. So even though these regulations have been around for some time, it even the regulations, it mentions historical controls. So this might be used, as I said on the previous slide, of real world evidence. But it does point out that these are kind of used in special circumstances, which I won't go into right now. The next one is the adequate selections of patients. So basically the patients in your study should have the disease you're trying to study. So how that relates to real world evidence, you have to be able to select the patients that have the disease or condition you're interested in. You have to be able to identify those patients. Assigning patients to treatment and control groups that minimize bias. So the regulations say that's typically done by randomization, but it doesn't have to be done by randomization. So again, this is sort of an opening to sort of non-traditional studies. Adequate measures to minimize biases on subjects, observers, and analysts. Typically, that's done with blinding. It's not the only method that's needed. It's not. Other methods are needed as well, but typically, this is where blinding comes into play. And in real-world data, blinding may not be possible, so that's something to keep in mind. But it's not a requirement. We know traditional studies often, well, sometimes don't blind. Well-defined and reliable assessment of subjects' responses. So again, that has to do with outcomes. You need good outcomes, and that's something we'll talk about further in this talk. And finally, adequate analysis to assess the drug results. So that's basically where one of the, that's basically our area in statistics, to make sure we're doing the correct analysis, the appropriate analysis to come up with valid estimates. So that's what an adequate and well-controlled study is. And I tried to emphasize how it might relate to real world evidence. So the effort in real world evidence sort of got a kickstart in 2016, well, I guess 2017, with the 21st Century Cures Act. There's a little discrepancy. The title says 2016 and this bottom says 2017, so I don't know which is correct. So 21st Century Cures was a major piece of federal legislation. It did many things, and one of the many things it did was address real world evidence. And it called FDA to establish a program to evaluate the potential use of real world evidence to help support the approval of a new indication for an already approved drug, and also to help support or satisfy post-approval study requirements. So two things here. It basically is focusing on already approved drugs. The feeling is it's hard to get real-world evidence on a drug that's not already in the market. The second thing about post-approval studies, it's explicit of what that might mean, but it could probably mean post-market safety studies, post-market confirmatory studies in the context of accelerated approval, or maybe post-market pediatric requirements. Now, importantly, 21st Century Cures says that none of this is going to change the evidentiary standard. So on the last slide, I talked about what an adequate and well-controlled study is, and nothing about that is going to change. In 21st Century Cures makes that explicit. The bar for evidence is the same. So if you're going to do something in real world evidence, it still has to satisfy existing laws and regulations. So in order to address 21st Century Cures, FDA had a plan for a number of guidances, and that's those guidances are sort of spelled out in a 2018 FDA framework on real world evidence. And there was a lot of progress in 2021 on getting draft guidances out. These are listed in chronological order. So starting with real-world data, assessing electronic health records and medical claims. Let me try to use the cursor. And I'm going to skip to the third for a moment, real-world data, assessing registries to support regulatory decision-making. So these are two parallel guidances that basically address a fit-for-purpose of two sources of real-world data. Here, electronic health records and medical claims data, and here, registry. So they address like validating variables from the data set, documenting, doing QA, QC procedures and documenting that. But they're two basically parallel guidances on different sources of data. And they're what we call agnostic to the study design. Whether you're doing a randomized study or an observational study, the concepts in those two guidances apply. The second guidance has to do with data standards for real-world data. Now, for those of you who submit data to FDA, know that it has to be submitted by certain standards, and those standards tend to be CDISC standards. It's not required by law that FDA chooses CDISC, but that's what FDA has chosen, and that's what this guidance has chosen for real-world data. Now, CDISC is not particularly suited necessarily for real-world data, so there's a lot of transformations that have to occur, and this guidance addresses like advice on doing that transformation and documenting it. Finally, on the FDA guidances is this considerations for real-world data and real-world evidence. It's a very interesting guidance. I highly recommend you review that. It concerns both interventional and non-interventional studies, but the bulk of it actually has to do with the non-interventional studies. And it makes a few important points. Well, it makes a lot of important points, a few of which I'd like to emphasize here. It talks about study integrity a lot, that things have to be pre-specified. We're used to that in the interventional world, but it needs to be done in the non-interventional study as well. And in particular, some of the data sources may already exist while you're designing a study. And it's important to not review the data that would lend itself to any sort of biases. Now, FDA accepts that you may have to review the data to see if you have sufficient number of patients, a sufficient number of events, but a lot of care is needed that any sort of review of the data doesn't kind of imply that the study would be biased, and that should be documented as well. So it's a subtle thing there, something probably we could talk a lot more about, but won't have any more time, won't have the time to go into depth here. The last point of that guidance I'd like to bring up is the FDA's expectation that that sponsors will submit patient-level data. Now, that's the regulations for traditional studies, and that's going to be the expectation for real-world studies as well. And that may be complicated because sponsors may not own the data, the real-world data, so there might be additional agreements that are needed to be put in place to submit that data to FDA. I am less familiar with the EMA's guidances in this area. Actually, they have a guidance on registries. I guess similar to FDA's guidance on registries. The draft I guess it's final now. The draft has been around for a number of years. I have read that one. It's a very good document. And they also have their network data standardization strategy document that came out last year. Okay, in FDA's guidances and framework for real-world evidence, these two concepts, relevant and reliability, and this addresses the fit for purpose of the data. And these are concepts we pick up in our paper that Weili submitted, that's been in submission now. And Weili will flesh these out in that semi-quantitative approach that she'll talk about at the end of this presentation. She'll add a third category as well. But the two categories sort of to measure or to consider for real world data fit for purpose are relevance and reliability. Reliability, I'm sorry, let me start. Relevance is, does the data have the elements that you need? So can you identify what drug they're taking? Can you identify the outcomes, the covariates? And are there sufficient number of patients in the data set for your study? And along with that, is there sufficient follow-up perhaps? So does it have the data that you need? Reliability is how good that data is. And that includes concepts like accuracy. Does it measure what it's supposed to be? Completeness, how much missing data there is. And these concepts of provenance and traceability. Can you trace your data back from the analysis data set back to its ultimate source? And is that documented? So these are kind of the overarching concepts for fit for purpose data. Okay, now let me briefly go through each of the sections in our paper, some briefer than others. So the first section in our paper after that introduction is some examples of uses of real world data. So these are basically actually study designs. We wanted to provide a context so people who haven't seen this before understand how you might use real world data in actual designs. And we have here a spectrum of designs as well. I mean, this is not a long section, but just brief mention of a few real examples. Fully observational, single arm intervention with external control, and an example of a randomized study. The one I'm going to talk about today is based on a recent approval last July of a drug called Prograf. So this was sort of an important kind of milestone in real world data, real world evidence. It was a fully observational study that was used to provide a new indication. So FDA approved Prograf, that's what I just said, based on an observational study of real world evidence of effectiveness. So Prograf is a drug used for organ transplant. The new indication was to prevent organ rejection for those receiving lung transplantation. It had been previously approved for other organs, other solid organs, including liver, kidney, and heart. And it was actually used widely for lung transplantation off-label, which kind of made it suited for this real-world evidence because there was data out there in the world on its use for this new indication. The data source is this US Scientific Registry of Transplant Recipients. This is a data source that contains, or supposedly contains, all lung transplants that happen in the US. I'm not sure, it may contain other transplants as well, but for this purpose, it contains all the US lung transplants. So it's rather extensive. It's also a high quality data set. There's a protocol for how the data should be entered, and there's quality checks of its reliability, to use that word. For this purpose, it was supplemented by Social Security data to get information on mortality. So the patient compared patients in this registry to historical control in preventing organ rejection. Now, this historical control wasn't an explicit group of patients, but the sort of well-known, well-documented natural history that if you're not getting an immunosuppressant, you're very likely going to have organ rejection and likely death, or very likely death. This is, well-known kind of historical natural history. So the patients who were receiving the drug in question, Prograf, were compared to like what things used to be like before immunosuppressive. Now, naturally, there's been advances in medical care, but the effect is so dramatic that without a drug like this, you're going to have almost all organ rejection. And with this drug, if I recall, something like 90% or more of lack of organ rejection. So it's a dramatic improvement. So based on that, FDA determined that this sort of was a adequate and well-controlled study with fit-for-purpose data. And to approve a drug, you usually need not just one study, but multiple studies, or one study plus what we call confirmatory evidence, which comes from some other source. And so this study with the registry data was the adequate and well-controlled study, and the confirmatory evidence came from previous indications of this drug for other solid organs. So there was a similar sort of mechanistic clinical relevance of the RCTs that were studied for other solid organs. So that's the Prograf story. So I think, as I said, it's a big milestone, I think, in RWE. Okay, so let me move on probably quicker to some of these other elements. First, data integrity. So data integrity, overall, it's like, it's the data, the quality of the data. And there's, a few concepts that, some of which we've already talked about, the provenance. Can you, trace the data back to its origin? So in order to document integrity, you have to understand the provenance, and there might be, or likely will be, some sort of checking, like in traditional clinical trials, that the data are reliable, going back to the source data. Then there's the concept of curation. This curation is more complex than in traditional sources because the data weren't collected for these research needs. In a traditional study, the data are collected specifically for your study. Here, the data were collected for other reasons, and there's a lot of steps to get them into a form and analysis-ready data set. So that involves like cleaning the data, transforming, linking, documenting, and checking. And there was a Duke Margolis workshop in 2018. There's a slide at the end on references that discusses all the different aspects of data curation, and what are maybe thoughts on best practices in that area. Another concept that's relevant here is reproducibility and the documentation related to reproducibility. So, the word pretty much is self-explanatory. If you use the same methods on the same data, you should get the same results. But the documentation helps others understand the study. As this quote, well, not quote, but this concept from this paper by Wang and others, if there's enough information documented to reproduce a study, there's also a lot of information to evaluate the study. So the reproducibility kind of adds to the overall understanding and perhaps integrity of the study. That's a concept we talk about in our paper. Two other things here that we talk about, it looks like three other things we talk about under the section on data integrity or data standards. And I already discussed that earlier when I talked about the FDA's new guidance. So the next thing is data ownership and privacy. So, in a traditional clinical trial, you have informed consent. So the participant, by their right, understands exactly where their data or how their data will be used and agrees to that. But these RWE sources may not have that. That's a separate issue that I'm not qualified to speak on, is when informed consent is necessary and when it's not in the non-interventional world. But you have to ensure, that you do follow legal and, ethical issues when you make use of data. And sometimes that might involve some anonymization. And then there's a balance between, as you can imagine, there's a balance between how much you anonymize to protect patient privacy and the information you need to actually do your study. So you may have come across sometimes like the date of an event cannot be in the data, or the month of the event cannot be placed in the data, or the year of the event is not known. And sometimes the month might be sufficient, sometimes it's not. So there's kind of a balance here between this privacy issue and this kind of utility for a study. But ultimately you have to abide by legal and ethical issues. So a data source may not be sufficient for your needs. And as I mentioned this before in the regulatory considerations guidance but I think it's so important that I am going to say it again. Sponsors should document all access and analysis of the data and demonstrate that the choice of the final data set did not introduce biases into the study findings. Easier said than done, but as this field develops, maybe best practices around that will be better understood. So ascertainment of key variables. Now, the key variables are, defining your population, basically your disease definition, treatment exposure, what drugs or therapies they're taking, outcomes, covariates. Covariates might be used to create subgroups, or also in a non-randomized study, might be used to control confounding. But these are the key variables, and then this gets back to the relevancy of your data. Does it contain these key variables? And how do you get these variables from your data set? The data sets weren't designed for these research purposes or your specific study objectives. So there are various methods to extract this information. The first is expert algorithms. This may be simple or may be complex, but using the information of the data, for example, ICD codes, how do you define outcomes? Using prescription data, how do you define how many days somebody took a drug or so? Then there's machine learning. What a machine learning algorithm is used to extract the data, and I'm most familiar with maybe the supervised setting of that, where you have some actual outcomes that you actually know the correct answer in some cases, and you train the algorithm on that to be able to identify the outcome. In either expert algorithm or machine learning, text mining may come into play. So you could base things on specific fields, but there also might be additional information in more open fields like doctor's notes. So there might be some need for text mining of more open fields, either in the expert algorithm or in machine learning. A single data set, even as we saw in Prograf, may not be sufficient. In Prograf, you also needed the Social Security information to get the mortality. So you may have to link multiple data sets, and there are deterministic and probabilistic versions of that, and our paper will have discussions and references on those. Then there's the concept we discuss in this section called informed presence. And this is kind of a concept related to missing data for real world data. Now, the point is that the usual approaches to missing data, or the usual way of thinking of missing data in clinical trials, may not apply in this setting. Things in the real world are collected for a reason, and they may not, if they're not there, there might have been no need to collect it. So for example, a patient with diabetes is likely to have more glucose testing done than a patient without diabetes. So the patient without diabetes you don't think of them as having missing lab values. It was never really intended to be collected. So the actual presence of certain information is itself informative. I mean, you still have traditional missing data where something was supposed to be collected and is not in your data set. But there's kind of this broader concept of what's in your data set and what's not, and how you make use of that involves careful thought. Okay, the last topic that I'll be talking about is validating variables and assessing bias. There are several types of biases. The three main ones are confounding, measurement bias, and selection bias. Since our paper deals with the data, we're primarily concerned with measurement bias in the paper. But both confounding and selection relate to the data as well. For confounding, if your data set doesn't have important confounders, that's relevant. That's relevance. That's a lack of important variables. So it's kind of like presence or absence of variables for confounding. Similar for selection. You know, if you have a lot of loss to follow-up, you may have selection bias. But as I said, we're mainly concerned with measurement bias, or for discrete variables, misclassification bias. So there are, you know, two ways of thinking about it. I mean, there's two classes of this. Non-differential bias occurs in both treatment groups, and differential bias occurs sort of unequally between groups. And both types are bad, and they have different consequences, or some of the same consequences. But generally, the non-differential bias is gonna give you biased things towards the null. There's pathological examples where that's not the case. And biasing things to the null might not be a good thing if you're trying to identify a safety signal, or you're trying to, you know, establish the efficacy of a drug. But it's kind of, I think, a little better understood. You know, whereas differential bias, all sorts of things can happen. Validation studies try to get at the reliability of your variables, you know, typically comparing it to some gold standard. This may be done like kind of once for a data source, like someone has developed an algorithm for US Medicare data for myocardial infarctions, and they do a validation study and come up with certain parameters like sensitivity, specificity, and positive predictive values. And then somebody who does another study may not have to redo the validation study, or it may have to, in certain cases, be kind of data set dependent, studies dependent. So some sort of validation is necessary. Whether a special study needs to be done is case specific. And that special study may be a sub-study of your overall study, or sort of a completely external study. Okay, so this last thing I'm going to talk about is a little bit about quantitative bias assessment. There are many forms of this that have been around for a while, like what's the bias from an unmeasured confounder. That concept has been around for more than 50 years. The people have formulas for that. There are formulas for measurement bias. If you know the sensitivity and specificity of your measurement, what's the effect on the estimate. So those are sort of assessing bias, maybe the direction, the magnitude of the bias, and uncertainty. What's relatively new, and not brand new, but you know, maybe in the last 20 years, is like putting it all together, taking all the biases and figuring out, you know, what the effect is on the magnitude and the uncertainty of that bias. And you know, it involves expert judgment. There's no doubt about it. It's not like it comes out of nowhere. You have to make a lot of assumptions, but the value of this is it makes clear what assumptions are necessary, and then you can argue about what those assumptions are. So I think it kind of clarifies the whole approach, and probably goes a step further by just saying, well, this effect size is so big it couldn't be explained by bias. This kind of takes it a step further. Okay, so with that, I'm done with my portion. I'm going to turn it back to Weili for the last topic and some conclusions.
Thank you very much, Mark. Next slide, please. So Mark actually set up a really nice stage on this portion of the presentation on proposing a semi-quantitative approach to assess fit for purpose real-world data sources. So as Mark mentioned, that in assessing the fit for purpose or fit for use real-world data sources, we can evaluate data relevancy, reliability, but we also have another dimension in terms of regulatory issues. So in terms of data relevancy, as Mark mentioned, that we could identify key variables to see whether or not a certain real-world data source has collected these certain key variables, because we can now evaluate all the data points, right, collected in real-world data sources. But what we can do is to identify the key important variables, such as disease condition, treatment exposure, outcome variables, confounders, etc., and then evaluate if the data source relevant for answer a certain research question. So basically, relevancy reflects in the representative of data to target population, key variables information, and we also want to evaluate if a certain RWD sources has a sufficient size of the patients. We will describe more in the next couple of slides. Data reliability is on the ascertainment and the processing of information, and accuracy and consistency in the data ascertainment, and also completeness and the missing information. Regulatory information or issues, as Mark mentioned, in several of these recent FDA draft guidances related to the data transparency, traceability, auditability, and also data format and standard, as well as how the real-world data should be submitted for regulatory considerations. Next slide, please. So in our paper, we propose a semi-quantitative approach to evaluate the different aspects of this data relevancy or reliability. One have to really note that if you think about it, in evaluating a fit for use, fit for purpose real-world data sources, it's actually very much research question, disease treatment specific, because of the detailed algorithm actually need to be developed specific to the research question and the disease condition. So what we provide is actually, or propose is actually a guiding principle on how one could do such assessment. So, and then once we propose that guiding principles in actual assessment, if we have several, say, options for real-world data sources to be evaluated, we'll be able to use a similar algorithm and concept in evaluating those data sources and to see which one is actually more fit for purpose for use for your research question. Next slide, please. If you think about it, again, in the real-world data sources, there are probably hundreds of thousands of variables, data points to be collected, right? What Mark described is sort of like steps towards what one need to do in terms of using employing advanced analytics approaches like expert of algorithms, natural language processing, machine learning to ascertain those key variables for a real-world data source. And then to evaluate either validating the key data points and also assessing any biases of these data measurements. And then we can come to this point in assessing fit for use. So some of the assumptions we make is basically your disease conditions can be identified through ICD-9 or ICD-10 codes, could clearly identify that disease of interest for your research question. And also outcome variables, when you answer the research question, it's on the disease, a certain patient, the disease population for a specific outcome interest that a treatment may have an impact on, and then leading to an outcome specific to a time frame. So the response and outcome variables can also be clearly defined and meaningfully ascertained and also meaningful to patients and physicians. And the treatment exposure for the research question is clearly defined. For example, drug A versus drug B for that comparative effectiveness research question, hypothetically. And then with observational research, because of non-randomized nature, we need to really minimize bias and confounding if the research question is not just descriptive, exploratory in nature, but there is involved a confirmatory hypothesis testing. There might be a list of confounders. You could work with your collaborators, such as clinical physicians, to identify a key confounding variables to be ascertained to answer this research question. And then there's always a study duration, a follow-up time frame that is of interest to answer your research question that you could also assess. We need to know that for a real-world data sources, it can be very high quality. Basically, it's really ascertained without much, measurement errors, misclassifications, etc. However, it still needs to be relevant to answer your research question. If not, the data source is still not fit for purpose. Next slide, please. So what we propose is actually what we call the semi-quantitative approach to assess fit for purpose real-world data sources. The first dimension is on relevancy based on specific research question. So when we talk about relevancy, as Mark mentioned, there are a few key variables we could assess on the relevancy in terms of, say, disease population, right? Just imagine that you have a RWD source and you want to see if that RWD source is can be used to answer this disease question, research question on, for let's just hypothetically for patients with AML, blood cancer, what is the drug, the treatment effectiveness of treatment A, at 12 months of after 12 months of treatment, something like that. Then you should be able to define the disease condition for these patients in the RWD data sources, and then calculate the number and percentage of subjects having that disease condition. So that's also related to sort of sample size. If you have like 1,000 patients RWD source, but only like 10 patients have that disease condition, then it's not a good data source for you to use. So you can actually do a stepwise assessment. If you only see 10 patients when you need 100 patients to answer this research question, then probably you need to move on to another RW sources. And you also want to define your response outcomes, and you can actually calculate, the number and percentage of patients with such a response outcome. One caution is that if your research question is on comparative effectiveness research for regulatory purposes, you want to really do this sort of assessment blinded to the study team, so that only a small group of people can know the outcome assessment, because otherwise it's potentially biases you sort of research and study integrity also. Treatment exposure, definitely that's something you could determine how many patients are exposed to drug A versus drug B as well, so that you could be adequately know what your comparator would be, and if that data set is fit for purpose for that particular research question. As we mentioned before, for a certain research question, there is also a certain list of confounding variables that you could ascertain, and then you could also basically calculate, the percentage of patient subject information on these key confounding variables. Time frame is always important, right? Because with a certain research question, it also have an element of time frame to achieve that research objective, so that assessing the time frame will be important as well. Next slide, please. So the second dimension is on the reliability assessment. So reliability speaks to several sort of aspects. One is quality. So the quality really speaks to the validity of the data elements, the logical plausibility of data. For example, we have a certain, gender, age variable has a certain possible range, right? If you have somebody with age 200 years old, that's probably wrong. And data consistency for a patient with related data fields. Then I will say one example is, if you have a BMI measurement, which is about 40, for example, whereas for that particular patient, the weight and height actually is all normal, then the BMI may be calculated incorrectly. So those are kind of things that one could assess, make assessment of. And also definitely, sort of like a data standard, etc. Actually internal standards or external data models that you can reference as well. Completeness, as Mark mentioned, is one is for missing data, whether or not the certain data fields is missing based on the clinical, the real clinical care setting. But there is also a sort of a concept of informed presence or informed non-presence, meaning that certain patient information may not meant to be collected at that clinical visit. So we have to make that sort of assessment. I think another deeper sort of element, aspect on the completeness is on the data density, actually. So what it means is we could assess for a particular real-world data sources how often patients visit their clinicians or primary care physicians. Basically, how often they are being followed up. We could sort of standardize that kind of measurement by, say, the average time of patients being visiting their physician, have an encounter per, say, 1,000 patient year of follow-up, that kind of measures. Another one is how often patients' lab information or tumor measurements being ascertained based on the real clinical care setting. So that can also be measured and then standardized per sort of like per 1,000 patient year of follow-up, that kind of a measure. and exposures too, and also confounding variables, how often it's captured, etc. So those are all can be calculated based on the sort of like your research questions and how relevant those measures might be. Next slide, please. Another aspect is what we call the generability score. What it means is to really describe your real world data source and how representative that data source and the subject in the data source representative of the background disease population, like epidemiology. So what it means is if you have a real world data source, on AML patients, and we could compare the common elements of this data source with the epidemiology of the background rate, for example, for AML. If the background or the on average general AML patients having the disease probably is say 65 years old, but in your data set, the average patient age is 40, then definitely there is some differences, right, between your RWD data source versus the sort of epidemiology of the disease. There's also age range. For example, the gender, say the gender ratio. If there is a gender ratio not 1-to-1, but maybe 2-to-1, for example, male and female, then you sort of you sort of data sources have a little bit different ratio. Then that's also reflect that your real world data sources may not have as much as external validity as you would like. Next slide, please. This third dimension, what we introduce is fit for research. That's really can be assessed qualitatively instead of quantitatively. So the reason FDA draft guidance also talk about data provenance, traceability, and the format, especially requirement for CDIX standard for use of real world data sources for regulatory decisions and the data curations, etc. Those can be assessed, documented qualitatively as well. Next slide, please. So with that, I will just conclude with a few concluding remarks. As you all know, that real world data and real world evidence is evolving rapidly and is playing an increasingly important role in clinical development. Just thinking back, 4 years ago in early 2018, I really, at least personally, I felt that I knew very limited information in this space. But now that, with the working group and a lot of other interest groups working in this area, we just feel like a huge inroad has been made in terms of both, with other disciplines, but also statisticians playing a major role in this field. So again, we feel like, quantitative quantitative scientists definitely can play a critical role in conducting the real world evidence research. And also, we could help really lead in that process in answering research questions and conceptualize the research objectives, study design, assessing data fitness, and proposing analytical approaches, and interpret results and communicate to the general public. And our statisticians can really take a forefront role in shaping up in this field, in the design, analysis, methodology research, and in guiding the team for the execution of those research plans. So with that, thank you very much for the invitation and for your attention. Thanks. Yeah, we have some selected references as well, as you can see on the screen. Vivian?